Welcome to this explainer on ocular thermodynamics. Today, we're exploring something that honestly sounds a bit like science fiction, but it's actually an everyday reality in modern medicine. We're going to look at how doctors deliberately manipulate the fundamental physics of temperature and energy transfer, literally just to heal the human eye. I know, it sounds crazy, but it's happening every single day. So let's just dive right into this, because it begs a deceptively simple question, right? How exactly can extreme temperatures be safely harnessed for medical treatment? We are essentially setting up this fascinating battle of fire versus ice, and it's all happening right inside one of the body's most intricate, delicate, and vital organs. Here's our roadmap for today. 1. Energy in the eye. 2. Heat, gradients, and blood flow. 3. Freezing with cryotherapy. 4. Heating with thermotherapy. And 5. Thermodynamics in action. Let's get started with Section 1, Energy in the Eye, the basics of light and heat. We've got to start with a really foundational rule of physics here. Energy cannot simply disappear. When electromagnetic radiation, specifically light between 400 and 1400 nanometers, enters your eye, it doesn't just vanish into thin air. It gets converted into mechanical, thermal, or photochemical effects. Now, for our purposes today, we're zeroing in on that thermal component, where optical energy transforms directly into heat. And the absolutely fascinating part is that the eye isn't heated uniformly. Different biological pigments act like these targeted heat sinks. Melanin, which is located right at the back of the eye, absorbs a really broad range of visible light. Xanthophyll, concentrated right at the very center of your vision in the macula, well, that soaks up blue light like a sponge. And hemoglobin in your blood absorbs blue, green, and yellow wavelengths. Because of these distinct biological absorption profiles, the distribution of heat is highly specific to the exact tissue being targeted. This highly specific absorption kicks off a really elegant two-stage process for retinal heating. First up, the optical energy is absorbed. In fact, roughly half of all the irradiation energy entering the eye is absorbed right within that back layer. Then, stage 2 kicks in. The thermal energy diffuses away from that central absorption site and spreads outward into the neighboring retinal tissues. And it is absolutely crucial to understand why this diffusion matters. Think about it. If the heat didn't diffuse, the concentrated energy would just instantly burn a microscopic hole straight through the tissue. Moving on to Section 2, Heat, Gradients, and Blood Flow, the Physics of Spread. That outward diffusion we just talked about, it creates what physicists call thermal gradients. A thermal gradient simply describes a difference in temperature across space. Because the center of the targeted spot becomes the hottest, heat is forced by the laws of physics to spread outward toward the cooler surrounding tissue. It's kind of like touching a hot stove. A microsecond tap confines the burn to the very tip of your finger, right? But if you leave it there for a full second, the intense heat spreads through your whole hand. The exact same thing happens in the eye. Time is a critical variable here. Short laser exposures, we're talking under 100 microseconds, they confine the energy tightly to the absorbing region, because the heat just literally doesn't have time to diffuse. But longer exposures, over 1 millisecond, well, they allow that thermal energy time to spread into neighboring areas. So doctors aren't just controlling the power of a laser, they're actively controlling the timeline of the heat spread. Now, here is where it gets really cool. That highly vascular layer at the back of the eye acts just like a car engine's radiator. As blood flows through it, it doesn't just deliver oxygen and nutrients, it physically carries thermal energy away. So your eye basically comes with its own built-in liquid cooling system, just like a high-end gaming PC or a car engine. This continuous perfusion actively cools the eye down, acting as this massive, intrinsic protective mechanism against severe light damage. We can really see the clinical stakes of this energy absorption when observing something like the sun. A normal 3mm pupil causes a totally safe 4 degrees Celsius rise in retinal temperature, but dilate that pupil to 7mm, letting in vastly more light, and you get a massive 22 degrees Celsius rise. To put that into perspective, that is the difference between a warm summer day and literally boiling water. It crosses critical thermal thresholds, turning what would be completely reversible heating into irreversible permanent tissue damage. Let's look at Section 3, Freezing with Cryotherapy, the Power of Ice. This is where ophthalmologists bring out specialized tools called cryoprobes to deliberately pull heat out of the eye. Now, we need to clearly distinguish between careful cryopreservation, which preserves cells by gently slowing down their metabolism, and what we're actually talking about here, which is cryocoagulation. If you use a cryoprobe to extract thermal energy too rapidly, the intracellular water simply doesn't have time to gracefully escape the cell. Instead, it freezes in place, forming these jagged ice crystals that literally tear the cells apart from the inside out. It's a calculated, deliberate, and highly destructive use of extreme freezing. But, that destruction is actually exactly what we want for healing. In a process called retinal cryopexy, the cryoprobe extracts heat until the retina turns completely gray-white. This causes a very controlled tissue injury and targeted inflammation. Then, over the next 1 to 3 weeks, a strong scar forms. Usually we think of scars as a bad thing, right? But in this case, a scar is exactly the goal. It acts like biological superglue, permanently welding a torn retina shut. And because this freezing is applied externally to the outside of the eyeball, surgeons can effectively use it even when things like dense cataracts or hemorrhages block the clear path of a traditional optical laser. But remember our radiator analogy from earlier? That built-in liquid cooling system is constantly rushing warmer blood into the treatment area, aggressively fighting and opposing the cooling process. To overcome this, vitreoretinal surgeons actually have to press the cryoprobe firmly against the globe. Imagine having to literally squeeze a thick radiator hose shut just so the engine can finally freeze over. That's essentially what the surgeon is doing here, using physical brute force to locally cut off that warming blood flow so the underlying retina can ultimately freeze. Oh, right. Section 4, Heating with Thermotherapy, the Power of Fire. Flipping to the exact opposite end of the temperature spectrum, let's explore the power of fire. This is where light energy is deliberately absorbed by ocular pigment and converted into intense, concentrated heat. The biological effect of this heat depends entirely on the temperature reached. At about 60 degrees Celsius, the relatively weak molecular interactions holding cellular proteins together simply begin to break down. The proteins literally unfold and denature. Think of how the clear, liquid part of an egg turns solid, opaque white the second it hits a hot frying pan. The tissue effectively cooks, resulting in an immediate, visible whitening of the retina, starting cellular necrosis. But if you push that thermal energy even higher, up to roughly 100 degrees Celsius, the game changes entirely. At this point, the water in the tissue completely vaporizes. This leads to immediate, catastrophic, and complete tissue disruption, which is exactly why managing the dosage and the duration of these lasers is so incredibly critical. It's actually fascinating how perfectly the thermodynamics of heating mirror the mechanics of freezing. Optical energy enters, the local temperature rises, proteins denature, and controlled necrosis occurs. Whether you're freezing or heating, crossing specific, calculated thermodynamic thresholds results in a carefully controlled, localized injury that ultimately heals or stabilizes the overall structure of the eye. And thermal therapy isn't just about maximizing destruction anymore. Modern techniques are brilliantly nuanced. Take selective retina therapy, for example. It delicately warms the retinal pigment epithelium, that back layer we talked about earlier. This warming prompts the release of biological factors, basically jump-starting the eye's natural healing response without ever having to visibly burn or destroy the tissue. Or consider transpupillary thermotherapy, often called TTT. This is a long-duration, low-power approach used to slowly bake small melanocytic tumors. And because doctors know TTT can have a high tumor recurrence rate if used all by itself, it's frequently used as a really powerful adjunct alongside focal radiation. So doctors are deliberately manipulating subtle levels of heat to trigger precise biological responses, all while minimizing any collateral damage to healthy vision. Which brings us to our final part. Section 5, Thermodynamics in Action, two sides of the same coin. We've looked at fire, and we've looked at ice, but the absolute biggest takeaway here is that both laser photocoagulation and cryotherapy operate under the exact same fundamental laws of physics. In laser therapy, electromagnetic energy becomes thermal energy, and heat naturally diffuses outward into the tissue. In cryotherapy, thermal energy is extracted, and heat flows away from the eye towards the colder metal probe. Both techniques are simply deliberate manipulations of thermal gradients. They both dictate exactly how fast energy moves, exactly how far it spreads, and exactly how long the local temperature is altered to achieve a miraculous therapeutic result. So, knowing how precisely extreme heat and freezing cold dictate cellular survival, I'll leave you with this question to think about. Do you view the human eye as merely a biological organ or a dynamic thermodynamic system just waiting to be harnessed? It is absolutely incredible to consider how an intricate understanding of simple physics allows doctors to turn potential sources of profound injury into incredibly precise, life-changing tools. Thanks for joining me for this explainer. Keep questioning, keep learning, and I'll catch you next time.